was to search for risk factors for multiple sclerosis, and I will try and give a little summary of what we're doing in our group, and also actually some of the groups we're collaborating with. But it will only be some of them. So I thought I would start with saying a little bit about who I am, and then why am I interested in looking into multiple sclerosis. And then I'll go through risk factors divided up into genetic risk factors and lifestyle risk factors, and also how they interact with each other, and also a little bit about new ways that we are approaching looking at heterogeneity of multiple sclerosis. So who am I? Let's see if we can get oops, never mind. So I grew up in Skåne, on a farm. You can see the picture there. Not always sunny, and at the moment there's lots and lots of snow there. Then I did my undergraduate studies in natural sciences and molecular medicine in Cambridge University at Union College. And Union College is, I think today, we should give them a little extra hooray, because they were one of the first colleges, the very first college in Cambridge that allowed women to study to academic degrees. So International Women's Day today, we should really think about Union College and many other colleges that paved the way, making it possible to study academically for women as well. And that was not very long ago. It was in the beginning of the 1900s. So it's not, it feels like it should have been much earlier. But anyway. Then I did my PhD in 3 places: Lund University, and Washington University in Seattle, and also Karolinska Institute. And why did I move around so much? Well, I followed my supervisor. He happened to be moving from job to job. So I ended up in 3 different places. And after that, I did postdoc both here at KI and also Oxford University in UK. All of that was looking into the genetics of type 1 diabetes. So since 2005, I've changed focus, and I'm now looking into multiple sclerosis rather than type 1 diabetes. But the techniques are the same. A lot of the factors are even the same, so it's rather similar. I'm married to Alistair here, who doesn't usually wear such hats, but sometimes. And I have 2 daughters, Emmy and Lily, and 2 cats, Ayla and Jura. And when I don't work, I sing in a gospel choir. So tonight, despite my voice, I'm supposed to have a practice. And also I do some needlework when I get time off. So then why multiple sclerosis? Well, the main reason why I moved from type 1 diabetes to multiple sclerosis is very boring. That's because the funding ran out and there was an opportunity in MS. But there is another reason too, why I get motivated at least to carry on. And one of them is this person here called Fraser Shaw. He used to work in a bar in an island called Islay outside Scotland. My husband comes from Scotland, so we often go there. And I've met him every year from 2011 till 2015. And he had MS, and he got progressively worse. He was diagnosed in 2011 when the first time I met him. And then he got worse and worse and worse every year we saw him. And he died in 2015, not because of MS directly, but because of the treatment he's been given. So he was treated by one of these new treatments called natalizumab, which is very effective for most patients. I don't think it was so effective for him. But he had a severe side effect, which is when a virus called JC virus spreads in the brain and basically destroys it. And he died from that because he wasn't diagnosed early enough. But he was, while he lived, he worked in a bar, but his main interest was music. He was a folk music player, and also he wrote lots of music. And he started the Islay Music Festival, or the Islay Sessions, which are still running every year. So he had a very quick disease course from 2011 to 2015, really bad. Then I have another friend, a much closer friend. She's called Eva, and she has made this picture and many others. She was diagnosed in 1996. She's the same age as me, 50 years old. And she has very mild symptoms and has most of the time not been treated at all. Now she's on treatment, but most of the years she hasn't had no treatment at all. It still affects her life. She still feels, she has sensory problems in her legs, she has vision problems on and off, and she feels depressed. Whether that's because of MS or not, I don't know, but it quite likely could be. But in her case, since she got diagnosed, she's changed career. She used to be a secretary at Ericsson. She's now trained to, is working as a priest in the Swedish church. So she's been able to do quite a lot in her life after this diagnosis. So it's really quite different how the disease affects a patient. And that is, to me, very curious. And of course, you would like to help those where it goes really bad, like Fraser. So what is MS? MS is, the name comes from, means numerous scars, and you can see in these pictures, actually I should really work out how to work. There are signs here, white dots that come and go. These are serial MRI images of the same patient, and you can see that there are white dots coming and going. And these are scars where the myelin has been destroyed. And what you see is an enrichment of water, actually. That's what's visible here, but it's due to myelin being destroyed. So we think that what happens is that the immune system attacks the myelin that surrounds the axons in the brain, and they go away, and when they go away, then they function a bit like the plastic around an electric cable, so that they insulate, so that the current can go there, or the signaling along the nerves itself, and that becomes much worse when the myelin is gone. We don't know exactly what immune cells are involved and how it happens, but we think that it's cells from the periphery that for some reason have cross-react to myelin structures, and we don't know what they are, and that they come in from the blood vessel, enter through the blood-brain barrier, and starts to destroy the myelin sheet. There are a lot of things that happen. As I said, it's very different how the patients feel when they have, or what the symptoms they have, but they often come in bouts. So you often get an attack, and then you get better if you have the measure of how bad you feel along here. And then you often get back to almost the same as before, but not quite, and then you get a new bout, and it gets worse and worse. And then eventually you get to a stage where you get progressively worse even without these bouts. These bouts happen when there is a new scar forming in the brain, but there are many more scars forming than bouts. So it seems to matter where the scar is forming. So for some of them, they will affect you so that you actually notice the symptom, but for many of them, most of them, you can compensate. You have lots of other cells in the brain that take over. Another thing that happens during your disease course is that you have brain atrophy, which is much more rapid than for all of us. We all have brain atrophy as we go older, but for MS patients it's much quicker. And the result is that a lot of different functions are affected. What we usually think of, I think most of us, when we think about MS, it's a patient that can't walk or has problems doing things with their hands or so. But the most common symptoms is actually problems with vision and also depression. Another interesting thing regarding the impact, which I was aware of fairly recently, was how the patients rate their life. So at the top there's a scale of how well they feel. It's quite subjective. But if you look at the top line is the general population with different age groups, and the lower line is the MS patients. And MS patients who are the age of 18 to 29, they feel as bad as 80-year-old healthy individuals, which is really quite dramatic. That's, of course, on average. I think Fraser clearly would have fallen into that category, and I probably wouldn't. So it depends on if this is on average. Also, MS affects the employment rate, which you have in the lower graph there. So as you have had MS longer and longer and get more disabled, the rate of employment reduces. And it reduces really quite early on before you have symptoms that are really not that bad. It's also, you can see that there's quite a difference between different countries in Europe, and luckily in Sweden we're doing better. So patients seem to be functioning better even when they are getting worse. So that was a little of why study MS. It's a rare disease. I have friends that I can see a direct application I would like to do something. Let's go into the genetics, which has been my focus in my research. So, what's the evidence that genes are involved at all? This is not a monogenic disease that you have families where the mother has it and then the daughter and then her child in turn. This is a multifactorial disease where lots of genes seem to be involved, and there's only a slight increase in risk. So if you look at monozygotic twins, the concordance is around 25%, and it's much lower for dizygotic twins. And of course, dizygotic twins share much less genes, so that would then argue that genes are probably involved. And as you go down this list, the further down you go, more distant related the individuals are, and the lower the concordance between the pairs. And using that data, we can estimate the heritability, which is, in this case, 64%. So it explains some of the why you get MS, but not all. There are also other factors that explain why you get MS. We have been involved in a series of studies looking for genes. These are studies you can't do as one research group. You have to collaborate. So this is work done by the International MS Genetics Consortium. Basically consists of all MS groups, almost all MS groups interested in genetics in the world. And this particular study, which is not actually accepted yet, although it's published by archives, was doing a meta-analysis between all published genome-wide screens, which was about 14,000 patients, and a little bit more controls, and then replicating that in a new cohort of 40,000 MS patients and 30,000 controls, which we contributed quite a lot to from Sweden. And the plot is a circular plot of the genome, so at the top you have chromosome 1, 2, 3, 4, etc., all the way around until you come back up to chromosome Y. And there are 200, more than 200 actually, regions that are associated. Those are the black bars that go up here. That's the evidence, the p-value. The higher, the more significant, although it's cut here, so you don't see all the really significant values. So 200 regions, of which the major one is in the HLA region on chromosome 6. And most of the genes seem to be involved in the immune system. Previously there's been a debate, but it could be maybe primarily a neurodegenerative disease, not an immune disease. But the genetic association seems to point towards the immune system being the primary risk factor or risk function. In the HLA region on chromosome 6, there are many alleles and genes involved. There's a list here. And these genes are involved in antigen presentation, which all of you know, to T cells. So they present peptides, so it's quite logical for an autoimmune disease, or what we think. We haven't proven it's an autoimmune disease, but we think it's autoimmune. That antigen presentation is involved. So the main associated allele is the RB1 which has an odds ratio of about 3. And then there are several other alleles of the same gene that are associated, and also some at other genes. DRB1 is a class 2 gene, HLA gene, whereas the others are here at the bottom are class 1 genes. I'll come back to that. What we've also seen is that some of these factors interact with each other. So over here we have interactions with two different terms, two different alleles that together affect the risk. So it's quite complex, and this is just the HLA, then we have 200 other ones. So a lot of genes. One way you could look at it is looking at the effect size, so how much does each gene increase the risk of MS, and here's a plot of, not quite all, but some of them, in order of the odds ratio. And as you can see, the ones with the lowest odds ratio is just 1.1. It's very little increased risk, so then maybe you wonder who cares. But if it pinpoints to what function they are involved in, then it might be really interesting, even though the increased risk for each individual one is very little. But as you can see at the far end, the odds ratios go up a little bit. So if we zoom into those and we color the ones that are the HLA region red, then you see that almost all of the ones with the highest odds ratios are from the HLA region. And those brackets up there, they're supposed to indicate the factors that interact with each other. So there are some interactions as well. We'll come back to this picture adding lifestyle exposures to it as well. Okay, so we've identified more than 200 genes. How much of the heritability have we explained? Heritability was 64%, and of that, 18% is explained by the HLA region, or MHC region, 9% by the others, and we have 73% left to explain. So we haven't done so well, really. Although we do understand some of the functions that are involved now, which we didn't before. So why could it be that we have failed? Well, it could be that there are many, many, many genes with very, very small effects, and that the sample size, although we were looking at 40,000 cases and 32,000 controls, that wasn't big enough. It could also be that some variants that affect the disease are rare, because our studies so far focused on common variants. It could also be that we estimate the risk for each variant wrong, so when we work out how much they contribute to the heritability, that estimate is wrong. And that could be because of interactions between genes. And I've already showed you that there are some, there could be many more. And what we think is also going on is interactions with environmental risk factors, which could then also affect epigenetics. Could also be that we overestimate the heritability of the disease. That used to be the case, but I think with the study I showed you at the beginning here, that is pretty close to the real estimate. So I think that is probably not likely explanation anymore. So if we then turn to environmental risk factors, what evidence are there for them apart from the heritability estimate that their environment is likely to play a role? Well, there are other studies showing that migration affects risk. So if you move from a high-risk country to a low-risk, or from a low-risk to a high-risk, you adopt the risk in a new country if you move early in life, if you move as a child or a teenager, but not if you move later. And there's also differences in risk in different geographic regions. We live in a region with a high risk, and closer to the equator it's low risk. That could either be due to genes or environment, because of course we have different genes set up as well. And as you saw, the concordance for monozygotic twins was 25 or so, so there's a lot of discordance for monozygotic twins. There must be a lot of other factors affecting the risk. The ratio of females to males is changing over time. There's many more females affected with MS than males, but the ratio is also changing, so that could be some environmental factors. And also we know from other studies that the immune repertoire is largely driven by non-inherited factors, so then that's likely to argue for lifestyle factors. So we have several different studies that we are involved in, trying to address both the genetics and the lifestyle exposures. And these are big nationwide studies, the GEMS, the INS, and the AIMS. The mega one is a local one, where we collect blood samples, and we also ask the patients and matched controls to answer questionnaires regarding their lifestyle. So questions like how much do you smoke, how much do you drink alcohol, what infections have you had, what other diseases do you have, what you work with, what have you been exposed to at work, and what is your education, and lots of questions. And the GEMS study was a study of prevalence cases in the MS registry a few years back, where we contacted all we could get hold of. Once for all. The AIMS study is an ongoing study of newly diagnosed MS patients throughout Sweden. And the INS studies are studies of patients treated with certain different disease-modifying drugs, which we are following more carefully. And as we're doing that, we're also getting the DNA samples and questionnaire data from them as well. So in total, we have now samples from more than 11,000 cases and 12,000 controls, most of which have been genotyped by now, but I don't have the data quite yet. Soon. And looking at the data from the questionnaires, which is work that's largely been done in Lars Alfredsson's group, we have identified many different factors that seem to affect the risk. And they're listed here. I will go into details for smoking, some details for smoking, EBV, and BMI. So all of those are risk factors for MS. But there are many more. Night shift work, drinking coffee and drinking alcohol is protective, so that's good. Being out in the sun is also, so go out in the sun, not today maybe, some other day, and take your coffee there. Don't smoke. Okay, so this is just some examples. So looking at the smoking. So if you compare the risk of getting MS for ever smokers, people who have smoked sometime, with those who never smoked, there is an odds ratio of 1.5. It's a bit higher for those who are currently smoking. And if you stopped smoking more than 5 years ago, then you have reduced your increased risk for MS. So it's good to stop smoking if you don't have MS. The more you smoke, the higher risk you have. Not surprising. Even if you're exposed, if you don't smoke yourself and you're exposed to passive smoking, that also increases your risk, which is a little worrying maybe. But you can sort of combat that by using snus or soft, moist snuff. That reduces the risk of MS. That was smoking. Then EBV, Epstein-Barr virus infections, that has for a long time been associated with increased risk of MS. So if you have had infectious mononucleosis or a severe EBV infection, you have an odds ratio of almost 2. And if you have antibodies towards a certain protein in the virus particle, then you also have an increased risk. And there's a certain region of that protein that seems to have an extra high risk. And we actually now have a potential autoantigen that cross-reacts with that, so we're very excited. But I'm not going to show you the data. That's one of the first autoantigens, so it's really exciting. BMI is an increased risk if you have high BMI when you're in your teens or 20s, but not your current BMI when you get the disease diagnosis. And that's been shown both by us in the AIM study and also in a study, much bigger study of NAS's health study in the US, and it's exactly the same result. So the higher BMI, the higher the risk, but it is during your around 20 years of age. Now you could, of course, because we haven't measured the BMI for the patients when they were 20 years old, we've asked them how much did you weigh at certain times in your life. And of course they could remember that wrong. So maybe we can't trust it. So one way of getting around that is to do something called Mendelian randomization. If you know that there are genetic factors that affect a certain trait, BMI, then you could see if those SNPs that are associated with increased BMI are also associated with MS. And if that's the case, that would be a stronger argument that BMI really is a risk factor. To do that, you have to have various assumptions. First of all, you have to have the assumption that the genetic variants are only associated with BMI, a modifiable risk factor, and not with any other risk factor for the disease. And you also have to make the assumption that they're not associated with the confounders and that they're not directly associated with disease, only through the modifiable risk. And that, of course, we don't know if it's true, but doing that, we've done that for BMI with a genetic risk score. So if you have a high genetic risk score for BMI, then you have an increased risk of MS. And that's been done both in our study here in Sweden and also in a study in the US with very similar results. So that seems to sort of strengthen our belief that BMI really is a risk factor. So if we then go back to this plot, now we add in the environmental risk factors. Now you can see most of them have quite high odds ratios. That is probably because our studies have been fairly small compared to the genetic studies, so we haven't been able to detect factors with small effects. There are probably lots of environmental factors that have small effects, but we can't see because we don't have big enough studies. Another thing we do is to look at interaction between risk factors. So we try to see if you are exposed to one risk factor. No, if you're exposed to two risk factors, do you then have a higher risk than the sum of being exposed to one or the other? And if that's the case, that argues for the two risk factors being involved in the same sufficient cause, the same group of risk factors leading to disease in the same group of individuals. And we've done that with HLA, so this is just to remind you that we have lots of HLA alleles associated, but the main ones are DRB1, 1501, and A0201. Class 1 and class 2. And we've looked at those two in relation to smoking. And what we see then is that if you smoke, then you have an odds ratio, and don't have DR15, you have an odds ratio of 1.5. If you smoke, do not smoke but have DR15, you have an odds ratio exactly about 3. If you have both, then the odds ratio is almost 6. And if you add the two individual exposures together, you see that there's a bit missing, a bit more to be explained, and that's the evidence of interaction. So this would argue that these two factors are involved in the same sufficient cause for disease. And we've done that, extended it to AO2, and that also interacts with smoking. Both of them interact with smoking, and they also interact with each other. So there's a three-way interaction. So all these are involved in the same sufficient cause. How might this work? We don't really know, but we speculate that smoking probably affects antigens post-translationally in the lung, and that you then induce a cross-reactivity to some existing self-protein in myelin, and then you get an autoimmune attack. At least that can explain the DR15 part. The AO2 is a little harder because it's class 1, but maybe it could act through a T receptor suppressor function. Same thing has been seen for RA in myositis, but the HLA alleles are different. So the HLA allele seems to control which disease you get, but smoking is a risk factor, increasing the immunity in all cases. If we summarize all of these, we've done this in all sorts of combinations of HLA interactions, and you can see there's lots of them that interact with HLA, which argue that these factors are actually involved in the immune system rather than in any other way, increasing risk events. And you can view it then here. Here I've put brackets for all the interactions, now in purple, and we can build them as these sufficient cause pies. We start to build them together. It's still patching, we still have lots to understand, but we're beginning to get there. Very quickly, just a few more slides. This new approach is to try and address heterogeneity. So the question is, why does someone like Fraser go really bad really quickly, and someone like Eva trundles along quite well over a long period of time? We are involved in, or be coordinating a big EU grant where we are addressing exactly this, where we will look both at genetic risk factors, both for risk for MS, but also for various outcomes of MS, so how severe you are, and then try and build risk groups within this and pathways, so that we can find ways of finding biomarkers to identify patients belonging to these different groups, so we can pinpoint which treatment they are given in the future. That's the idea. We'll see if we get there. It's a little far-fetched, at least for 5 years. We should probably have longer to do it. One of the standard measures for severity is something called the EDSS scale. This is just to remind me to say that we are using this, but we are also using very many new ways of measuring severity, including what the patient feels himself, which hasn't been done before, which I think is quite exciting. And all this can be done because of the Swedish MS registry, or neuro registry, so we can follow basically all the patients in Sweden through that registry, assuming they're given consent. And one risk factor we've already looked at when it comes to how fast you convert to this disease, where you have a steady progression in your disease, is looking at smoking. And but if you carry on smoking after you got MS, you can have an increased risk of converting faster. So it's a good idea to stop smoking to prevent you from getting MS, but it's also a good idea to stop smoking to make you not get such a severe disease once you have it. So that's it, but there are many people to thank, at least of all Thomas Olsson, Lars Alfredsson and Jan Hillert, who have been professors here for a long time at KI and have built up this platform of patients and the MS registry that we now can harvest from. And then there are some of the students who do both in the studies, funders, and at the bottom, the two international consortium that I'm working with, IMSDC and Multiple Sclerosis.